Thank you. I'd like to thank the organizers for allowing me to come and speak a little bit today. My name is Pat Madden. I am instructor in Shane Crotty's lab at the La Jolla Institute for Immunology. And I'm going to talk to you a little bit today about some of our work in germline targeting vaccinations and some of our recent successes in eliciting HIV broadly neutralizing antibodies in non-human primates. So in 2023, nearly 2 million people worldwide were still infected with HIV, and this really put the 2025 UN/AIDS target out of reach. And in fact, it's now 2026, and we did not hit this 2025 UN/AIDS target. And due to recent developments in sort of global health, this number is likely to actually raise in the coming years in terms of the number of people that were infected. And there's been incredible progress in the development of antiretrovirals for HIV prevention that should hopefully have a huge impact on the global epidemic. But most people believe that a safe and effective HIV vaccine is still needed to truly end the epidemic. Such an HIV vaccine would need to induce multiple classes of HIV broadly neutralizing antibodies against HIV. So many such broadly neutralizing antibodies have been isolated from people living with HIV, and they target five main epitopes on the HIV envelope trimer. However, thus far, we have been unsuccessful in eliciting true broadly neutralizing antibodies by vaccination in humans or more outbred animal models, such as non-human primates. There's a couple reasons for this. There's some major hurdles for the induction of broadly neutralizing antibodies by vaccination. The first is that the broadly neutralizing antibody precursors are found at extremely low frequencies in the naive B cell repertoire. This is due to rare genetic features such as extremely long HCDR3s, as well as specific rearrangements that are rare. In addition, these precursors also have extremely low affinity for the HIV envelope proteins, and they're easily outcompeted by non-neutralizing B cells during immunization and even infection. And even if these precursors, broadly neutralizing antibody precursors, can be primed in order to fully mature to true broad neutralization, the ones that have isolated thus far have extremely high V gene mutation frequencies, suggesting that they're going to be hard to elicit through vaccination. And therefore, new and novel vaccine strategies are needed to try to target these broadly neutralizing antibodies. So one method for HIV vaccine design uses these isolated broadly neutralizing antibodies to reverse engineer immunogens that can stimulate the naive B cell precursors that have similar sequence motifs to known broadly neutralizing antibodies, and therefore may be able to mature into those broadly neutralizing antibodies. This reverse vaccinology leads to the idea or the concept of germline targeting immunization, which relies on multiple distinct steps. The first is using a engineered germline targeting immunogen where specific mutations have been introduced that allows for the recruitment of a very specific naive B cell. So priming of that B cell. And then you have to use a series of immunogens to shepherd that precursor along using progressively more native or fully wild type HIV envelope trimers to sort of hold that B cell's hand and walk it along the maturation pathway, hopefully inducing somatic hypermutation, gains in affinity, and eventually reaching neutralization potency and breadth against a wide variety of HIV isolates. And then the final step of this would be conversion of those matured broadly neutralizing antibody B cells into plasma cells to confer long-term protection. So recently, a few clinical trials have shown that this concept is not only possible but highly probable in humans, where the broadly neutralizing antibody VRC01 precursor cells can be primed in about 97% of humans using one of these germline targeting priming immunogens. In 2024, we published the first demonstration of priming precursors that are similar to the human broadly neutralizing antibody BG18. So BG18 is a very broad and potent broadly neutralizing antibody isolated from someone living with HIV. This broadly neutralizing antibody targets the V3 glycan epitope of the HIV envelope trimer, and it relies on a long CDRH3 loop to penetrate the glycan shield and make interactions with the protomer, the HIV envelope protomer. It also makes extensive glycan contacts in that interaction. The potential precursors for BG18 class for this BG18 broadly neutralizing antibody are extremely rare in the human naive precursor or the human naive B cell population, and they're found at about 1 in 50 million naive B cells. They're extremely rare, and went on to show that these precursors that are nearly identical to the human precursors can be found in rhesus macaques, but they are found at even lower frequencies. So they're about 2 or 8-fold lower frequency in the macaques. So roughly 1 to 100 or 400 million naive B cells. Despite that extreme rarity, we were able to show that with this germline targeting priming immunogen called N332-GT5, we can successfully prime these precursor B cells that meet the specific genetic features that we care about, which I'll talk more about in a second, in these rhesus macaques. We can get them to compete in germinal centers. We can find them in the memory B cells. And when we produce those antibodies from those B cells, and do structural characterization, we can see that the precursors that we're priming in the macaques exactly mimic the structural binding mode of the prototype broadly neutralizing antibody. So this is great. This was the first real demonstration of the first step of this pathway, showing that we can in fact prime these extremely rare precursor cells. But that leads us to the remainder of this pathway. Can we take these naive precursor B cells that we can prime and walk them along this maturation pathway? So in order to do that, we need a series of immunogens, and that series of immunogens needs to be progressively more native-like. So that means that there needs to be fewer and fewer germline targeting mutations as you go along until finally you're immunizing with fully wild-type HIV envelope trimers. At the same time, you need to increase the envelope diversity that these B cells have seen in order to induce breadth. In order to attempt this, along with our collaborators at Scripps Research Institute and Bill Sheaf's lab, specifically John Steichen, we designed this large monkey experiment in rhesus macaques, where we used a series of boosting immunogens. Again, they're getting progressively more native-like. So you can see the prime has about 20 germline targeting mutations. We then immunized with boost 1 candidates with between 9 and 17 germline targeting mutations. The boost 2 has 2, and then we introduced fully wild-type trimers, and finally a cocktail of different fully native-like trimers. And these different immunogens sort of recapitulate the diversity seen specifically in the V1 loop of the HIV envelope trimer. Throughout this study, we take blood and lymph node fine needle aspirates to track the B cell responses in these animals. So our workflow for this is heavily reliant on the sequencing of the BCRs of these cells. So we use a sorting panel, which allows us to use both the priming immunogen and whatever the subsequent boosting immunogen is in our sorting. We sort the epitope-specific cells, so we use an epitope knockout to sort those specific cells. We also include the boosting immunogens, so we can get information by flow cytometry. And we also use an oligo hashtag on those boosting immunogens so that we can make bioinformatic binding calls after our sequence processing. So again, we're heavily reliant on the Immcantation workflow for doing our BCR analysis. And then we also use Seurat for gene expression analysis and also determining the thresholds for pro-binding. And in this study, the B cells that we care about, we term as BG18 type 1 BCRs. And to annotate those, we have a definition. And that definition is that they have to use the IgHG 341 D gene in the correct reading frame and position. They have to have a CDRH3 that's at least 19 amino acids or longer. And they have to have this specific residue at the D+2 position. So you can see the rhesus macaque IgHD 341 gene aligned here to the BG18 inferred germline. And so we primarily look in the memory B cells, and the data I'm going to be talking about today is primarily at the 2 weeks post each boost memory B cell time point. So when we just look at the frequency of these BG18 type 1s after each boost, we were extremely excited to be able to see that we can, in fact, detect these BG18 type 1 cells after each of the boosts out to 82 weeks and 5 boosting immunogens. And in our 3 boosting groups, groups 2, 3, and 4 here, we can see that 12 out of the 18 animals have persistent BG18s out to this late time point. And then again, using our bioinformatic boost binding call, we can show that at that time point, the majority of these BG18 cells are in fact interacting with wild type HIV trimers, suggesting maturation. We can then use ChangeO to collapse these BG18 sequences into clonal lineages, and then we can track their persistence over time. And in this case, what we did is plotted the frequency of the BG18 type 1 clones in each animal at each boost time point. So if you just focus down here at the bottom on group 4, you can see that between 4 and 8 BG18 type 1 lineages are primed in each of these animals. And in the case of group 4, every animal can carry at least one of these lineages throughout the entire study. And in fact, for the majority of them, those sequences are found at every single time point. So you can see some where there are 2 lineages that make it all the way to the end of this boosting study, where in some other animals, they're just single lineages. But what's really cool about these lineages is that they're not just persistent, they're incredibly expanded. So this purple lineage here in this animal RQD20, we have thus far isolated over 3,000 BCR sequences from the memory and GCB cells that belong to this single lineage. And so we next wanted to use IG5mel to reconstruct the lineage trees from these clonal families, as we have previously used IG5mel in some of our previous publications. We did run into some issues where these extremely large clones, we have a hard time getting IG5mel to run. If people have thoughts or tips about that, I'd be glad to hear that. But in order to use IG5mel on these clones, we had to do some down selection of sequences. And I'm going to just show you 2 lineage trees here, one from this clone and one from this clone. So on the left here, we have this extremely large clone of 3,000 sequences that we've now down selected to 500. And you can see an incredible diversity of sequences that are making it to the end. So if you just focus on the red dots here, which is our final post-boost 5 time point, you can see that there's incredible intraclonal diversity in this lineage. Here's another lineage that has a slightly different maturation trajectory, where there's some diversity early on and only one sort of clade of these early sequences seem to be able to get boosted, although we are seeing still intraclonal diversity. So you can see here that these lineages are accumulating a large amount of SHM. They do seem to be maturing. Does that translate into better functional data? And the answer to that is yes. So if we take sequences from these different animals, we create monoclonal antibodies and test their affinity over time to 2 different wild type trimers. You can see that after each boost, they're gaining affinity for fully mature wild type trimers that starts at about boost 2. We also get asked quite often whether or not there are other lineages that are not BG18 type 1s in these animals that we seem to be boosting. To answer that question, I went back into the sequencing data and just looked for persistent clones, so clones that can be found at multiple time points that are expanded, that have long CDRH3s, and that don't meet the definition of being BG18 type 1. So when we do this, there's actually very few clonal lineages that meet that criteria. However, we did observe 3 clonal lineages from 3 different animals that use the exact same V heavy gene. They use very highly related D genes, and 2 of the 3 use the same V lambda gene, suggesting that this is a new type of response that we might care about. And you can see that these are incredibly expanded. This top one, we've thus far been able to see 6,000 sequences from that clone. And we hypothesized that these are potential BG18-like or type 3 responses where they don't share the common genetic features, but they might mimic the structural binding mode of BG18. And so in order to determine that, our collaborators in Andrew Ward's lab solved structures of some of our vaccine-elicited antibodies. So on the left in orange here, we have the prototype broadly neutralizing antibody BG18. You can see the CDRH3 reaching down, making extensive contacts with this green glycan, as well as the protomer. Our 2 type 1 antibodies that we have structures of, you can see mimic that interaction almost exactly because we have a very strict definition of their D gene. And so you can see there's basically no difference in the tip of that HCDR3 and its interaction. And then this new type 3 lineage that we found that doesn't share genetic features, you can see also uses slightly different residues and slightly different D gene to basically almost exactly mimic that structural interaction, suggesting that this is a new type of a potential BG18 binding mode. And finally, we wanted to determine whether or not these monoclonal antibodies have cross-clade neutralizing ability. Are they really truly neutralizing antibodies? So we made monoclonal antibodies. We tested them on a small panel of 9 viruses and showed that after boost 3, we start to be able to see some small amount of neutralization. But after boost 4 and 5, we can see that these antibodies are now neutralizing 6 and up to 7 of these 9 viruses. So we took the best antibodies from post boost 4 and 5, and we ran them on a larger panel of 63 viruses. And we were pleased to see that we get breadth on this 63 virus panel of between 10% and up to 46%, which is incredible and some of the best um, broad neutralization induced by HIV vaccines thus far. So hopefully I've been able to show you through this data that we believe that this pathway is not only possible, but now highly probable in these animals, where we can prime these naive B cells, we can walk them along this gradient with these multiple boosting immunogens. We can see increases in somatic hypermutation, affinity, neutralization potential, and breadth. But the final piece of this puzzle is whether or not there's serum neutralization activity. So we looked at serum neutralization post boost 4 and 5, and we saw very little, if any, true serum neutralization after boost 4 and 5. So to try to increase this, we decided to give 2 additional boosts in a somewhat exploratory manner. The first was an additional cocktail of 4 trimmer immunogens of recently circulating HIV strains. And then finally, we gave them a cocktail of 4 nanoparticles in the hopes that the potential valency of these nanoparticles would increase the antibody secreting cells. And in fact, that's exactly what we saw, where we see an incredible increase of the serum neutralization in these animals. And in fact, this one animal has 86% breadth on this 63 virus panel with GeoMean ID50 values above 400. And this is the best serum neutralization that has ever been seen in an HIV vaccine experiment. And in fact, this animal is in line with the serum neutralization that you observe in people living with HIV that have true bnAbs in their serum. So we're incredibly excited about sort of the prospects of continuing to iterate on this vaccine design. So in summary, hopefully I've been able to show you that these rare BG18 type 1 B cells can be primed and shepherded from naive to neutralizing in non-human primates. More than 50% of the NHPs develop cross-neutralizing BG18s after 5 boosting immunizations. These lineages can expand and persist, and the vaccine-elicited BG18s mirror the binding mode of the bnAb. And we can observe broad neutralization in serum. In addition, the first in-human clinical trial testing this priming immunogen is ongoing, and we have seen some good preliminary data suggesting that these same precursors can be primed in humans. And hopefully, I've also been able to show that this germline targeting vaccination studies require robust high-throughput BCR analysis pipelines. And so we're incredibly grateful for the existence of Immcantation, and very happy to present this data here today. So with that, I'd like to thank Shane Crotty and members of the Crotty Lab highlighted here who have been very helpful. And then a big shout out to Bill Schieff's lab at Scripps, specifically John Steichen, who designs all of these immunogens, and then some of our other collaborators who help with this and our funding sources. And I'd be happy to take any questions. Thank you.